Prokaryotes have a very simple organization. In the E. coli, there are many genes under the control of a single promoter. That makes the whole process easier. Here we can see there are many genes which are controlled by a single promoter, and all these genes code for products which are related in some manner. For example, there may be enzymes which coordinately control a particular synthetic process or a breakdown process, and all the enzymes are together transcribed by a set of genes, and all the genes which are responsible for this type of associated polypeptides are under the control of a single promoter. The genes are transcribed to produce a polycistronic mRNA. Cistron and gene. The difference is genes are sequences of DNA that are transcribed into one mRNA, and cistrons are terms associated with mRNA. One cistron is a specific nucleotide sequence that translates into a particular polypeptide. So polycistronic mRNA means there are different cistrons. Each cistron is translated into one product. So in this mRNA molecule, there are many cistrons which produce many types of polypeptides. But the thing is, here all the polypeptides are related in the function. Therefore, the related polypeptides are under the control of a set of genes, and that is called as operon. And this set of genes are under the control of a single promoter. In the year 1960, Jacob and Monod published a paper in the Proceedings of the French Academy of Sciences demonstrating that two genes involved in lactose metabolism were coordinately regulated. The terms operon and operator were used in that paper for the first time. Since then, many types of operon have been discovered. The first operon was the lac operon in E. coli, and for their contribution, these two scientists received Nobel Prize in Physiology and Medicine in the year 1965. Operon refers to a group of genes or a cluster of genes which control the metabolism of a product coordinately or with cooperativity. Lactose metabolism is controlled by the lac operon, and all the genes and their controlling elements are a part of the lac operon. Some regulatory elements work from outside, and some are in the DNA included under the lac operon. Lac operon includes genes and their controlling elements which are required for the metabolism of lactose, the disaccharide. Gene expression means production of a product after transcription and translation of genes, and the lac operon has some structural genes which are associated with the metabolism of lactose. Lactose is a disaccharide, glucose is a monosaccharide, and for the E. coli cell it is easier to use glucose as the source of energy. So if glucose is available in the medium, the E. coli cell draw glucose and metabolize it to get energy. But if glucose is not available in the medium, or it is in very low concentration, in that case lactose is absorbed by the E. coli cells and it is metabolized by the bacterium. Lac operon can be either in on mode or off mode. It is activated when glucose is in short concentration in the growth medium or totally absent in the growth medium, and lactose is there in the growth medium. So the condition is no or low glucose and availability of lactose. So if the situation is like that, then only the lac operon will be in a switch on mode and the genes will be activated, 3 enzymes will be translated. Out of this, the enzyme beta galactosidase is the primary enzyme that cleaves lactose into glucose and galactose. For the entry of lactose from the external medium, the transporter protein is required, and that is beta galactoside permease. Third enzyme is beta transacetylase, and its function is it adds one acetyl group from acetyl coenzyme A to beta galactosides so that metabolism is efficient. This shows the cell membrane of E. coli. Lactose is outside in the medium. First, it will enter into the E. coli cell. For the entry of lactose, galactoside permease is required. Galactoside permease is the transporter protein. After entering into the E. coli cell, lactose breaks down to either allolactose or to glucose and galactose, and for this conversion beta galactosidase is the enzyme. Beta galactosidase is coded for by the gene Z and galactoside permease is coded for by the gene Y. The lac operon includes promoter sequence, operator sequence, 3 structural genes Z, Y and A. The controlling elements which lie outside the lac operon is the gene I that codes for the repressor protein. The gene I has a separate promoter. This promoter is independent of the lac promoter. Then there is cap binding site to which cyclic AMP and cap binds. Promoter is the sequence to which the enzyme RNA polymerase binds to start transcription. Operator is the target sequence for the lac repressor protein. Structural genes code for 3 enzymes: beta galactosidase, galactoside permease and transacetylase. Cap binding site is for the binding of the complex cyclic AMP cap. Let us have a look at the structure of the lac operon. I gene, it is the gene expressing the repressor, and this is the promoter for the I gene, and we can see it is different from the promoter for these three structural genes. Promoter sequence to where the RNA polymerase binds, operator to which the repressor protein binds, operator overlaps the promoter. This sequence is the binding site for cap cyclic AMP complex that is involved in the positive regulation of the lac operon. Operon can either be inducible or repressible. Lac operon is an inducible operon because the inducer, allolactose or IPTG, can activate it. On the basis of regulation, the lac operon is subjected to negative regulation, that is, the binding of a repressor represses the operon. It is also subjected to positive regulation because the binding of the cap CAMP activates or increases the expression of the lac operon. Let us discuss the negative regulation of the lac operon. Negative regulation means the repressor binds to the operator and prevents the transcription of three structural genes. During negative regulation, the promoter for the I gene is activated and the I gene undergoes transcription, then translation. As a result of the translation of the I gene, a repressor protein is produced. The repressor protein has a target sequence at the operator site, so the repressor protein binds to the operator site, and as the operator overlaps the promoter, RNA polymerase cannot bind to the promoter. That is, the binding of the repressor protein blocks the access of the RNA polymerase enzyme. As RNA polymerase cannot interact with the promoter, three genes Z, Y and A cannot be transcribed. So there is no transcription, no translation. And here the repressor is a allosteric protein. That means it can bind, it can dissociate when it undergoes conformational change. The conformational change of the repressor occurs when there is the inducer. Inducer here is allolactose, but IPTG can also serve as an inducer for this lac repressor. When the inducer binds, the lac repressor undergoes conformational change. In the new conformation, it cannot bind to the operator. As a result, it dissociates from the operator and the promoter gets free, the RNA polymerase binds, and there is transcription of these three genes. After dissociation of the repressor-inducer complex, the inducer is separated and the repressor is separated. That is the end of the negative regulation of the lac operon. Once the repressor goes out from the operator, the promoter site becomes free. RNA polymerase now interacts with the promoter. Promoter becomes activated and these three genes Z, Y, A are expressed and transcribed into a single polycistronic mRNA. This polycistronic mRNA is spliced at appropriate sequences and produces three enzymes: beta-galactosidase, galactoside permease, and trans-acetylase. CAP is the abbreviated form of catabolite gene activator protein, and it is a positive regulatory element of lac operon. CAP protein binds to another molecule, that is cyclic AMP. The level of cyclic AMP in the E. coli cell varies according to the concentration of glucose in the external medium. When the glucose concentration is high, cyclic AMP level is low, and low cyclic AMP cannot bind to CAP. It has very low affinity with the CAP protein. When the glucose level in the growth medium is low, cyclic AMP concentration inside the E. coli cell is high, and when the cyclic AMP concentration is high, then only it can bind to the CAP and it forms a complex with the CAP. So here we can see the binding of CAP and cyclic AMP, and this occurs when the cyclic AMP level is high. And once this complex is formed, then only it can associate with the CAP site on the lac operon, and binding of the CAP cyclic AMP complex to the CAP site activates the promoter very highly, and the expression of these three genes increase almost 50-fold, 50 times, so that more mRNA and more enzymes are produced. This facilitates the metabolism of lactose by the E. coli cell. Here is an overview of the regulation of lactose metabolism when it is in a on mode. So it is in a on mode means lactose is available and glucose is in short supply in the medium. And now what happens, the enzyme RNA polymerase binds to the promoter. Because promoter is free, the repressor has been dissociated. Then operator is free, we can see here, operator is free. And cyclic AMP CAP binds to the CAP site. This increases the expression of the lac operon 50-fold. That is why it is called as the positive regulator of the lac operon, and CAP is a positive regulatory element of the lac operon. So when RNA polymerase is associated with the lac promoter, repressor is not attached to the operator, and the CAP cyclic AMP binds to the CAP site. Three structural genes Z, Y, and A are transcribed to produce a polycistronic mRNA, and it contains three cistrons. One cistron is translated into beta-galactosidase, second cistron galactoside permease, and third cistron is translated into trans-acetylase. So we can say it is just a part of the E. coli genome. Until now, many such operons have been discovered, but the lactose operon is the first operon to be suggested by Jacob and Monod, And all the principles which guide the regulation of gene expression in prokaryotes is based on this lac operon. Lac is an inducible operon because it is activated in the presence of the inducer allolactose or IPTG. Lac repressor protein is the negative regulatory element that is coded for by the gene I, which is under the control of a separate promoter. When repressor binds to the operator, transcription is blocked and the enzymes are not synthesized. CAP is a positive regulatory element of the lac operon, and the negative and the positive regulatory element act in a concerted manner, means in a related manner, and this type of regulation of both negative and positive control is seen in the lac operon of E. coli. Thank you.